Hello everyone. My name is Kiran Chaudhary and I am your biology faculty at Infinity Learn by Shri Chaitanya. बहुत बहुत स्वागत है मेरी इस क्लास में स्टूडेंट्स। चलिए आज के लेक्चर की बात करते हैं। आज हम लोग क्या पढ़ने वाले हैं? आज का जो टॉपिक रहेगा इस क्लास के लिए वो है पैकेजिंग ऑफ डीएनए हेलिक्स। यस, तो आप सभी लोग जो मेरी इस क्लास में, इस लेक्चर के इस क्लास में पहली बार आए हैं, लेट मी टेल यू व्हाट वी हैव discussed till now before this lecture. So before this lecture where we will be discussing about packaging of DNA helix, we have already done the discussion on structure of DNA, right? और हम लोगों ने इसके साथ साथ में DNA का जो डबल हेलिकल मॉडल है उसके क्या कैरेक्टरिस्टिक्स है, वो किसने दिया और उसके क्या सेलिएंट फीचर्स है, वी हैव ऑलरेडी कवर्ड इन द प्रीवियस 2 लेक्चर्स। चलिए आज यहाँ से स्टार्ट करते हैं इस लेक्चर में कि डीएनए की पैकेजिंग किस तरीके से होती है सेल्स के अंदर। लेट्स स्टार्ट दिस सेशन। और सेशन स्टार्ट करने से पहले आई वुड लव टू गिव यू इम्पॉर्टेंट इन्फॉर्मेशन स्टूडेंट्स दैट यू कैन यूज किरण 40 कोड, KIRAN किरण 40 कोड का इस्तेमाल आप लोग कर सकते हैं टू गेट 40% off on the various courses that we offer at infinitylearn.com website. एक और important information जो आप सभी लोगों को समझनी है that we have started a new batch targeting NEET 2022 where we will be covering syllabus of 11th and 12th class, right? And the last day to enroll yourself for this batch for NEET 2022 full course is 25th of November. So hurry up and do not forget to apply code. Which code? Code Kiran 40. So Kiran 40 code को apply कीजिए and get straight 40% off. Get straight 40% off when you enroll yourself for this कोर्स, राइट? तो चलिए अब आगे की बात करते हैं, आज का लेक्चर स्टार्ट करते हैं। चलिए देखिएगा स्टूडेंट्स, सबसे पहले द वेरी फर्स्ट थिंग, हेलो सुलीन, वेलकम टू माय क्लास। तो आज हम लोगों को इस लेक्चर में सबसे पहले क्या काम करना है, बिफोर वी स्टार्ट अंडरस्टैंडिंग हाउ पैकेजिंग इज डन इनसाइड a cell of DNA helix, let us understand or let us try to calculate togetherly कि किस तरीके से how DNA or what is the exact length of DNA. So let us calculate the DNA length. तो चलिए स्टार्ट करते हैं। लेंथ ऑफ डीएनए फैक्ट से स्टार्ट करते हैं। अगर हम लोग लेंथ की बात करें और ह्यूमन सेल की बात करें, देखो बच्चों स्पीशी वाइज वेरिएशन आएंगे। Today in this class we will try to calculate the length of human cell DNA. और कौन सा ह्यूमन सेल का भी कौन सा टाइप है, डिप्लॉइड सेल की बात करेंगे। तो ह्यूमन का एक डिप्लॉइड सेल, व्हाट इज अ डिप्लॉइड सेल? डिप्लॉइड सेल मतलब उसमें डीएनए का कंटेंट 2n होगा। सो कंटेंट ऑफ डीएनए का मेरे प्यारे बच्चों, कंटेंट की अगर मैं बात करूं तो डीएनए का जो कंटेंट है वो क्या रहेगा, 2n रहेगा, क्योंकि ये डिप्लॉइड सेल है। तो ह्यूमन के डिप्लॉइड सेल में जो डीएनए होता है उसका लेंथ इज 2.2 मीटर। दैट इज द फैक्ट। लेट अस ट्राई टू कैलकुलेट हाउ साइंटिस्ट और पीपल गॉट टू दिस नंबर, किस तरीके से ये 2.2 मीटर को कैलकुलेट किया गया है। चलिए साथ में मिलकर सीखते हैं। बट टू रीच this number 2.2 meter we need to have some prerequisite knowledge, right? तो याद करने की कोशिश कीजिए, जब भी आप लोग फिजिक्स में कोई न्यूमेरिकल सॉल्व करते हैं तो आपको कुछ गिवन कंडीशंस होती है, गिवन दिस, गिवन दैट, एंड विद द हेल्प ऑफ दैट, गिवन दिस एंड गिवन दैट, एंड विद द हेल्प ऑफ अ proper formula you try to get the unknown thing. So let us consider this 2.2 meter as of now as an unknown quantity. तो objective क्या है? To calculate what is the length of the DNA in a human diploid human cell. तो prerequisite given knowledge जो आपको इस कैलकुलेशन को करने के लिए चाहिए वो क्या है, ध्यान दीजिएगा। prerequisite is सबसे पहला डिस्टेंस क्या है, दो कॉन्जिक्यूटिव बेस पेयर्स के बीच में क्या डिस्टेंस है, कितना डिस्टेंस है, आपको ये पता होना चाहिए. सेकेंड थिंग रिक्वायर्ड इज हाउ मेनी बेस पेयर्स आर प्रेजेंट, हाउ मेनी बेस पेयर्स आर प्रेजेंट टोटल इन द एंटायर डीएनए, राइट. तो कितने बेस पेयर्स हैं और दो पेयर्स, और कौन से दो बेस पेयर जो ऊपर नीचे हैं, राइट, कॉन्जिक्यूटिवली प्रेजेंट है, जस्ट एक के नीचे एक. सो ये एक के नीचे एक जो बेस पेयर्स है इनके बीच में कितना डिस्टेंस है और टोटल डीएनए के हेलिक्स में कितने बेस पेयर्स हैं. दीज आर द टू इंपॉर्टेंट थिंग्स यू शुड नो. हाउ मेनी base pairs are present in the entire length of the DNA present in a diploid human cell, and second thing, what is the distance between the two consecutive base pair. That is the prerequisite knowledge required to calculate the length of the DNA inside a human diploid cell. Chaliye, let us do the calculation. Calculation is quite simple. What you need to do is you need to multiply the total base pairs. You need to multiply total base pairs with the distance between two base pairs. तो ध्यान दीजिएगा बच्चों, ये आपके डीएनए का एक सेगमेंट है, राइट. With this image, what I was trying to explain you, with this image मैं आपको ये बताना चाहती थी कि this is how the DNA look like, but this is just a segment of DNA, part of DNA, not the whole DNA. But you can get this image can give you a reflection of the entire length of the DNA. तो देखो DNA के अंदर होता क्या है. DNA में क्या है, consecutively अगर आप देखें DNA के entire length की अगर हम बात करें तो क्या है, DNA में base pairs है और दो base pairs consecutive, देखो इस base pair और इस base pair की ध्यान दीजिए, between these two base pair, these are what, this and this, these are two consecutive base pair और उनके बीच में distance कितना है, 0.34 नैनोमीटर, है ना. अब देखिएगा, अगर इसी तरीके से आपको ये पता चल जाए, anyhow you come to know that how many base pairs are present in the entire length of the DNA, and you already know that what is the distance between the two consecutive base pairs. It is pretty simple to calculate the length of the DNA by just multiplying the total number of base pairs into the distance between two consecutive base pairs. So that's how the length of DNA is calculated. So irrespective if it is human cell DNA or amphibian cell DNA or any kind of species you can consider, this is the basic rule. So irrespective of the species whose DNA length you need to calculate, the basics will remain same. You have to multiply total number of base pairs present into the distance between two consecutive base pairs. So see, if you, when you will multiply the total length of DNA equals to multiplying total number of base pairs, 6.6 into 10 raised to the power 9 base pairs are present. 9 base pairs, 6.6 into 10 raised to the power 9 base pairs are present in a diploid human cell. Inside the DNA of a diploid human cell, you have to multiply it with the distance between two consecutive base pairs. What it is? It is 0.34. But that is this, students, this is nanometer. So if you will straight multiply 6.6 into 10, बहुत ध्यान से सुनना, ये important बात है, important trick है, इस बात का ध्यान रखना. अक्सर generally students avoid this important fact. Please do not make this mistake. When you multiply 6.6 into 10 raised to the power 9 into directly simply 0.34 nanometer, you will get the length of the DNA in nanometer. But you don't want to get that. Why? Because you want answer in meters, right? So if you just simply multiply this, you will get answer or length in nanometer. But we want in meter, so we need to multiply this with 10 raised to the power minus 9. Why are we doing this? Because in 1 nanometer, there is 10 raised to the power minus 9 meter. So 1 nanometer equals to 10 raised to the power minus 9 meters. Hence 0.34 nanometer will be equals to, in terms of meter, 0.34 into 10 raised to the power minus 9. Now when you will do this calculation, you will get answer. You will get the length in meter. So the answer is, if you will calculate it manually or using calculator, whatever, it's up to you, it's your wish. The answer that you will get is 2.2 मीटर. तो बताओ बच्चों, समझ में आया कि ये कैलकुलेशन किस तरीके से होती है? Tell me, were you able to understand how we got this? Okay, so आगे देखते हैं. Next important thing that we need to understand is, देखिएगा स्टूडेंट्स, dimensions of typical nucleus. एक typical nucleus का dimension होता है 10 raised to the power minus 6 मीटर. यानी कि diameter of एक टिपिकल न्यूक्लियस का जो डायमीटर है वो 10 रेज टू द पावर माइनस 6 है, यानी कि माइक्रोमीटर ऑर्डर का है. यस, इफ दिस इज अ रिप्रेजेंटेटिव डायग्राम ऑफ अ न्यूक्लियस ऑफ अ टिपिकल न्यूक्लियस, लेट मी राइट इट अगेन फॉर यू. राइट, तो ये जो डायमीटर है वो 10 रेज टू द पावर माइनस 6 है, यानी माइक्रोमीटर ऑर्डर का है. तो देखो प्यारे बच्चों, ध्यान देना, बी वेरी अटेंटिव एंड नोट दिस पॉइंट. डीएनए का लेंथ कितना है? 2.2 मीटर. जिस न्यूक्लियस में इस डीएनए को रहना है, द साइट वेयर दिस डीएनए विल रिसाइड इनसाइड एन यूकैरियोटिक सेल. Inside eukaryotic cell, what is the place or position of DNA? It is concentrated inside a membranous structure known as nucleus. तो DNA को न्यूक्लियस के अंदर फिट होना है. DNA की लेंथ क्या है? मीटर में है, और न्यूक्लियस का डायमीटर -6 यानी माइक्रोमीटर ऑर्डर का है. Now just imagine how amazing the nature is. कितने बेहतरीन, कितने शानदार तरीके से डीएनए की पैकेजिंग की जाती है. इमेजिन, आई वॉन्ट एवरीबडी टू फील दिस. इट इज रियली अमेजिंग एंड एक्साइटिंग एट द सेम टाइम कि डीएनए तो मीटर में है, डीएनए की लेंथ इज इन मीटर, and DNA need to fit inside a structure whose diameter is of micrometer order, 10 raised to the power -6. Imagine how beautifully in nature DNA is packed that the length of 2.2 meter get fit inside a structure which is of micrometer order. बताओ, अब आप सभी लोगों को ये समझ में आना चाहिए that why it is important to study, to know and discuss packaging of DNA. So this line actually creates curiosity inside each one of us to know, to learn, to understand how DNA is packed, right? क्योंकि पैकेजिंग में कुछ तो शानदार, कुछ तो कमाल की बात हो रही है, वरना इतने से माइक्रोमीटर ऑर्डर के रेंज के डायमीटर वाले स्ट्रक्चर में मीटर लेंथ का डीएनए कैसे फिट हो जाता है. Isn't it interesting? तो चलिए पता करते हैं. Let us know, let us learn together how this packaging takes place. Let us see. तो चलिए स्टार्ट कहाँ से करेंगे? We'll start with prokaryotic cell and then we will see how DNA is packed inside a eukaryotic cell. तो बहुत आसान है, चलिए देखिएगा. पैकेजिंग डीएनए की किस में देखेंगे हम लोग समझेंगे एक प्रोकैरियोटिक सेल में. ये रिप्रेजेंटेटिव डायग्राम किसका है मेरे प्यारे बच्चों? इधर लिख देती हूँ. दिस इज अ प्रोकैरियोटिक सेल, है ना. तो देखना, ध्यान देना आप सभी लोग, एक प्रोकैरियोटिक सेल के अंदर डीएनए जो होता है किसी मेम्ब्रेनस स्ट्रक्चर से बाउंड नहीं होता. inside a prokaryotic cell, DNA is not bounded by a membranous structure like nucleus in case of eukaryotic cell. So, prokaryotic cell lacks all the membrane bound structures like nucleus, right, like mitochondria, like chloroplast, etc. So, क्योंकि प्रोकैरियोटिक सेल में मेम्ब्रेन बाउंड स्ट्रक्चर नहीं होते हैं, न्यूक्लियस नहीं होता है, तो डीएनए एक प्रोकैरियोटिक सेल के अंदर साइटोप्लाज्म के अंदर ही कहीं कॉन्सेंट्रेटेड होता है. और जिस रीजन में, द रीजन इन विच डीएनए इज कॉन्सेंट्रेटेड इनसाइड द साइटोप्लाज्म ऑफ prokaryotic cell is known as nucleoid or genophore. So, this is the term given to the region in which DNA is concentrated inside a prokaryotic cell. The name of the region is nucleoid or genophore. समझ में आया? Right? तो चलो आगे देखो. अब DNA के पैकेजिंग की बात करते हैं. तो DNA जो होता है प्रोकैरियोटिक सेल के अंदर पैक होता है, सुपर कॉइल्ड होता है. इट्स नॉट ओनली कॉइल्ड, इट इज सुपरली कॉइल्ड सो दैट इट कैन स्टे इनसाइड अ वेरी स्ट्रक्चर विच इज ऑफ रिलेटिवली स्मॉलर लेंथ. ओके, अब ध्यान दीजिएगा, हुज डायमीटर इज ऑफ रिलेटिवली लेस ऑर्डर इन कंपैरिजन टू द लेंथ ऑफ द डीएनए. तो ध्यान दीजिएगा, ये इस पैकेजिंग को करने के लिए या फिर इस सुपर कॉइलिंग को करने के लिए कुछ प्रोटीन्स यूज होते हैं, कुछ प्रोटीन्स हेल्प करते हैं. सो द प्रोटीन्स दैट हेल्प्स टू पैक और सुपर कॉइल डीएनए इनसाइड अ प्रोकैरियोटिक सेल आर नोन एज non-histone proteins. Yes students, and these non-histone proteins are basic proteins known as polyamines. तो यहाँ से दो बातें समझ में आती है. There are two important things that you should take from this board or from this slide. Two things. Histone proteins are not found in prokaryotic cell. That means histones will not help in packaging DNA inside a prokaryotic cell. Then who will do that? Who will help in packaging? Some other basic proteins will do, whose name is, for example, polyamines, right? And these are non-histone proteins. तो देखिए यही सब यहाँ पे लिखा हुआ है. तो ये हमारी प्रोकैरियोटिक सेल है और ये रहा हमारा डीएनए. प्रोकैरियोटिक सेल का डीएनए इज एक्चुअली कॉन्सेंट्रेटेड. इट इज कॉन्सेंट्रेटेड इन अ रीजन नोन एज न्यूक्लियोइड. कहाँ है हमारा ये रहा. ज़्यादा ऊपर जाने की ज़रूरत ही नहीं थी, मैंने नीचे ही आपको मेंशन कर रखा था. ये देखिए आपका, जस्ट गिव मी वन मोमेंट. तो ये देखो, ये आपका क्या है, न्यूक्लियोइड. तो जिस रीजन के अंदर आपका डीएनए कॉन्सेंट्रेटेड है प्रोकैरियोटिक सेल के अंदर, उसे आप न्यूक्लियोइड या फिर जीनोफोर कहते हैं. और नॉन हिस्टोन प्रोटीन्स हेल्प करती हैं जिनका नाम पॉलीअमीन्स है और वो बेसिक है, नॉन हिस्टोन टाइप की हैं, हेल्प्स इन सुपर कॉइलिंग ऑफ डीएनए. बताओ क्या आप सभी लोगों को यहाँ तक की बातें क्लियर है? कैन वी मूव फॉरवर्ड? ओके गुड. लेट अस सी व्हाट वी नीड टू अंडरस्टैंड नेक्स्ट. चलिए, ये यहाँ तक हमने बात करी कि व्हाट डिड वी डिस्कस. वी डिस्कस टुगेदर दैट how DNA is packed inside a prokaryotic cell. Now let us see how DNA is packed inside a eukaryotic cell. So definitely in comparison to a prokaryotic cell, DNA of a eukaryotic cell will be compactly arranged and also the packing will be complex, right? Who helps here? So DNA is compactly packed inside a eukaryotic cell. In that also, inside eukaryotic cell, the DNA is packed inside nucleus, which is a membrane bound structure with the help of special proteins known as histones, right? What are histones? Histones are a set of basic proteins, positively charged basic proteins. Why these are basic proteins? Because they are rich in basic amino acids like lysine and arginine, right? So I hope this much should be clear to all of you that DNA inside a eukaryotic cell is packed inside a membrane bound structure known as nucleus, and in nucleus who is helping to inside nucleus who is helping to pack DNA. These are special proteins known as histone proteins. Yes, Akash, sure. Let me know what is not clear. I would love to tell you. Again, let me know which line is not clear, Akash. I would love to tell you. Can you elaborate your doubt a little bit? What exactly you did not understand, Akash? Any line? Okay. For Akash, I would love to repeat this portion, okay, because I am unable to understand what are histones. Okay, perfect. So, Akash, listen this very carefully. What are histone proteins? Histone proteins are actually basic proteins, positively charged proteins. This is what the chemical nature of histone. So, the chemical nature of histone is they are basic proteins, they are positively charged proteins. Why they are positively charged or why they are basic proteins? Because they are rich in basic amino acids like lysine and arginine. You must be knowing from your chemistry knowledge that lysine, arginine amino acids are basic amino acid. Hence, the protein formed out of these amino acid will also be basic in nature, right? So, histone, talking about the chemical nature, it is a basic protein. This is the chemical nature. Now, let us discuss what is the function of this histone protein. Histone protein helps to pack the DNA. Remember, DNA is 2.2 meter long. DNA is 2.2 meter long, right? And the nucleus of an eukaryotic cell is of 10 raised to the power minus 6 meter order. Imagine, length of the DNA is 2.2 meter and this DNA need to be fit inside a structure known as nucleus whose diameter is of 10 raised to the power minus 6 meter order. Imagine how beautifully these histone proteins, how beautifully they will work to compactly condense the entire 2.2 length DNA so that it fits in a structure whose diameter is of 10 raised to the power minus 6 meter. Tell me, Akash, now this is clear or not? Let me know now your doubt is clear or not. Waiting for your response. Chaliye, tab tak let us see what is there. So, packaging of DNA in eukaryotes is quite complex because of length, obviously. Thank you, Akash. Okay, doubt of Akash is resolved. Chaliye, let us learn next. So, I hope this much is clear. Now, ab yahan par dhyan dijiye, please focus on this image. This is a representative diagram. What of whose representation this is? This is how DNA is compactly packed. DNA is compactly packed inside eukaryotes, inside the nucleus of eukaryotes. So, first of all, who will do the packing? Who will help? Histone will help. How exactly they will help? I will teach you on the next slide. But let us imagine how this packing is done. See, this is what? This is the DNA. Okay, double helix of DNA. Now, it will be packed to form next nucleosomes. This is what? This is what? Nucleosome. This is what? A complex packed structure of DNA. So, this is level 1 packing. Level 1 packing of DNA. Level 1 packing of DNA helix. So, level 1 is to form nucleosome. Again, nucleosomes will be further compactly packed to form solenoids. What is this? You can see. This is a solenoid. Again, solenoid will be compactly packed to form super solenoid. Super solenoid will be compactly arranged or packed to form chromatin. Chromatin during cell division will again undergo compact packing. stacking to form chromatids, and finally you will get chromosomes. So see how beautifully, compactly DNA is packed from double helical DNA to nucleosome, from nucleosome to solenoid, from solenoid to supersolenoid, from supersolenoid to chromatin, from chromatin to chromatids, and finally to chromosomes. Isn't it amazing? Isn't it amazing? Yes, do not worry, just wait for a few minutes. I would love to tell you, I would love to solve your doubt related to heterochromatin and euchromatin. It is there in today's discussion, but just wait for some time. चलिए आगे देखते हैं. So although all this thing I have already explained to you, let us try to revise it a little. What are histones? These are set of positively charged basic proteins. Next, they are rich in, why they are basic proteins? Rich in amino acid residues like arginine and lysine. Okay. Next thing, there are 5 types of histone proteins. Remember, these are set. This is a set. So there will be multiple types of histone present in a set. That is why you are calling it a set. So 5 types of histones are present, right? 5 types of histones is present. Histone H1, H2. H2 has 2 types, H2A, H2B, H3 and H4. So H1, H2A, H2B, H3 and H4. Now remember this very important point, that is H2A, H2B, H3 and H4 are actually present in a pair. So 2 molecules of H2A, 2 molecules of H2A, 2 molecules of H2B, 2 molecules of H3 and 2 molecules of H4 will finally form a unit made up of 8 molecules known as octamer. See here, these all, so let me write it for you, into 2, into 2, into 2, into 2. So how many? 2, 2, 2, 2. So 4 times 2, 8 molecules, right? So histone, unit of 8 molecules. So all these 8 molecules, all these 8 molecules will form a unit, a structure known as octamer of histone, also known as core of histone. See here, core of histone. So these 8 units will actually form this structure. This is DNA, right? This is DNA. You need to focus on this structure. This structure is known as core of histone, also known as octamer of histone, also known as new bodies. Okay, this is what histone that helps to form 4, right? H2A, H2B, H3 and H4 all will occur in a pair to form a unit known as histone, core of histone or octamer, right? I hope this much is clear. Now let us see how DNA will be packed with the help of histone proteins. So see, this interesting part is coming now. The negatively charged, DNA is negatively charged, right? And histones are positively charged. So I am pretty sure that with your chemistry knowledge, it should be clear that negative and positive will do what? Attract. So it is quite easy because of this opposite nature. Negatively charged DNA will wrap themselves around positively charged histone. So see here. What is this? This is the core of the histone. What is the core of the histone? This part. This part is actually core of the histone, positively charged structure. And what does DNA? What is this? DNA will wrap around this core of histone molecule. And what this H1 will do? Remember, we discussed together, there are 5 types of histone: H1, H2A, H2B, H3 and H4. H2A, B, 3 and 4 in a pair will form histone octamer. Then what was the function of H1, right? This should be a question, this should come in your mind. So let us discuss togetherly that what is the function of H1. H1 actually helps to seal the DNA that is entering and exiting. See, this is what? Entry. This is what? Exit. So H1 histones actually seals the DNA. So this DNA which is wrapped around this histone molecule actually stays together or stay there, get fixed there, get stabilized. So that's the function of H1 histone. So now this whole structure is known as what? Known as nucleosome. What is a nucleosome? Nucleosome will have histone octamer made up of 8 histone proteins, H1 histone which is sealing the DNA and a part of DNA. So histone octamer or core of histone, H1 histone and a part of DNA will form a structure known as nucleosome. Remember, we did the discussion. What is a nucleosome? Remember here, this is nucleosome. So here I showed you the representative diagram, and on that slide we were actually trying to find out how this nucleosome look like. So double helical DNA will be condensedly wrapped around histone octamer to form compactly arranged structures known as nucleosome. Two nucleosomes will be connected with the help of a linker DNA, right? So it is like a continuous thread where you can see beads like structure known as nucleosome. So now again nucleosomes will be further compactly arranged to form solenoid. Solenoid will be compactly arranged to form supersolenoid, then from that chromatin, then again chromatids, and finally chromosome. I hope this much is clear to all of you. Is there any doubt? Let me know. Is there any doubt? Yeah, it looks like bead-like structure. Very right, Chandramouli. Let us see the next thing that we need to discuss in today's class. Okay, and that's exactly what I was trying to explain over here, that nucleosomes are actually repeating unit of chromatin. When you observe a chromatin thread inside an electron microscope, you can actually see that chromatin looks like a thread in which there are multiple beads, right? So this bead-like appearance of chromatin actually shows that it possesses nucleosomes, and hence we can say nucleosomes are the units of chromatin, repeating units of chromatin. Tell me, is this clear? Thank you, Sunil. Thank you everyone for letting me know that. Chaliye, see, this is again DNA, double helical DNA. It will be condensed to form nucleosomes, and when you will see this structure, under an electron microscope, this is actually an image of an electron microscope where you can see beads connected with, lot of beads connected with threads, right? So what are these beads? Beads are nucleosome. Nucleosome is further compactly arranged to form solenoid, again supercoiled to form supersolenoid, then chromatin, then chromatids, and finally chromosome. So this structure you can see is actually of a chromosome. बताइए, समझ में आ रहा है? Yes, hello Killer Jack. Tell me, good evening, welcome to my class. Killer Jack, I remember your name is Jack, right? Okay, chaliye, next we need to understand few more important fact that chromatin material, although this whole discussion I have already done, but for the namesake I am explaining it again because it is there on your slide, but this whole thing we have already discussed, right? So what this first line is trying to say is nucleosome. If you say nucleosome is further coils to form chromatin, I have already explained you, right? Next is chromatin fibers are further coiled to, okay, you also remember this. I hope everybody should be clear with the order. Order is what? First double helical DNA, right? Then it will form nucleosome, then solenoid, then supersolenoid, then chromatin, then chromatid, then chromosome. So this line is trying to say that when you need to pack on higher level, above chromatin level, then there is a requirement of additional set of proteins known as non-histone chromosomal protein. So non-histone chromosomal protein will actually helps to pack more compactly chromatin material. So chromatin से ऊपर, beyond the level of chromatin, if you want to further pack the DNA, you require NHC protein. You require NHC protein. What are NHC protein? Non-histone chromosomal proteins, right? So अगर आपको chromatin level से ऊपर, beyond chromatin level, you want to pack, then what you need? You need non-histone, a set of proteins collectively known as non-histone chromosomal protein. With the help of these additional set of protein, NHC protein, you can form chromatids, and with that you can form chromosome. So that's exactly what is represented over here. So finally, from a double helical DNA, you have reached to a stage where you have so superly condensedly packed DNA inside a chromosome. Isn't it amazing? Batao bachcho, is there any doubt? Is there any doubt? Any doubt? I'm not, how about Jack letting me know what exactly is your doubt? I'm still not clear with your doubt, okay. So try to organize and put it so that I can in one go understand, okay. So try to work on your doubt a little more. So this is the thing that we need to discuss. This is the last discussion point of today's class, that let us understand the two regions that are there in chromatin. So the discussion will be on chromatin, and there are two regions in a chromatin, heterochromatin, euchromatin. And how this division is done? Division of chromatin into hetero and euchromatin is done on the basis of staining. How different regions of chromatin are responding to stain. Based on this, there are two regions or two divisions done of chromatin. One type of region seen in chromatin is heterochromatin, and another is euchromatin. So see, Chandramouli, now I am going to address your doubts. So please pay attention. Hello everyone. Okay, चलिए एक बार फिर से समझने की कोशिश करते हैं. See, so we need to differentiate heterochromatin and euchromatin on the basis of 3 points: staining, packaging nature, and transcriptional activity. So let us do a good comparison. Between the two. So let us talk about the stain. Heterochromatin takes dark stain, right? They takes dark stain. That means they get densely stained. So under a microscope, if you will see after staining, you will see chromatin, you will see that some regions you will see as dark region and some you will see as light region. So when you are looking chromatin under an electron microscope, the darkly stained regions are heterochromatin and lightly stained regions are euchromatin. So stain, if we talk about, euchromatin will take light stain. Okay, next is packaging. चलिए देखते हैं what type of packaging. Heterochromatin is densely packed, and in comparison to heterochromatin, euchromatin is relatively loosely packed, not entirely loosely. This loose and dense is relatively used word. So in comparison to heterochromatin, euchromatin is loosely packed. Okay. So packaging, if we talk about, it is heterochromatin is densely packed. Euchromatin is loosely packed. relatively, okay, in comparison to heterochromatin. Next, we need to understand is transcriptional activity. तो ध्यान दीजिए मेरे प्यारे बच्चों, what is transcriptional activity? So transcription क्या होता है? Transcription is actually a phenomenon during which from DNA genetic information is copied. Genetic information is copied into RNA. So when from a DNA, mRNA is produced or RNA is produced, that process is known as transcription. So if we talk about transcriptional activity, heterochromatin is transcriptionally inactive. It is transcriptionally inactive. It is transcriptionally inactive. Transcriptionally inactive. And talking about euchromatin, it is transcriptionally active, right? So euchromatin takes dark stain, it is densely packed and it is transcriptionally inactive. That means mRNA won't be produced from heterochromatin. Euchromatin takes light stain, loosely packed in comparison to heterochromatin and transcriptionally active. That means RNA will be produced from this part of DNA known as euchromatin. बताइए बच्चों, is this clear? Is this much clear? Tell me, is this much clear? Let me see if there is any other doubt. Okay, I can see. Good. Oh, Ram Nivas is saying good sister. Thank you, Ram Nivas. Okay, in human body 3m, 2m, 2.2 meter. That's the data given in your NCERT. You need to consider that. You need to use this whenever an option is given in your NEET exam. Okay. So next is okay, clear. As far as I can see, all the doubts are clear. Okay students, thank you very much for joining my session. If you like me and this session, give me a thumbs up and do not forget to subscribe Infinity Learn by Sri Chaitanya. Okay students, and also hit the bell icon so that whenever I will come to take class live, you will know and you won't miss any single class and any single concept that I will teach you. Okay, so Chandramouli is asking about translation. Do not worry Chandramouli, I am teaching you molecular basis of inheritance. So transcription, translation, everything will come in discussion. But for you right now, translation is the process where proteins will be produced from RNA. Genetic information will be copied to proteins. Okay. Okay, is that okay Chandramouli? Can we conclude the session now? But do not forget to join the session where I will teach you how translation of protein formation takes place. Okay students, thank you, bye bye, we'll meet again in the next session.